happen to your body? You are going to sweat, right? What is the purpose of sweating? To cool down your body, to maintain the temperature of your body. Is it so? So we are maintaining the internal environment. Our body temperature shouldn't rise above. What is the normal body temperature for humans? 37. Good, great. So our body temperature should not rise above, let's make a round down, like 40 degrees Celsius. Okay, and in doing so, our body maintains our body produce certain responses like sweats, or if the temperature is low, our body starts to shiver to generate more heat in the body to raise the temperature of the body. Okay. Okay, the very next thing is what else they do. uh, they evolve. The living organisms evolve, right? Do you have any idea about the evolution? Okay, what was the first organism on this earth? On this earth? Really? It's bacteria. Simple. So we say every organism is basically evolved from bacteria. 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 Okay? You must know. You might forget, but you must know. Okay. Okay, then the last one is they're made up of cells, which is their basic unit of body. And moreover, when we talk about cells, we should talk about the genetic code. Now, let's define genetic code. What is genetic code? It's basically a smaller sequence of DNA. Do you have any idea about the DNA? No? Yes, okay. So if this is the DNA, then a very small portion is known as gene. Each gene decides our characteristic, like we have genes for our hair color, hair texture, eye color, even the shape of an eye, our skin texture. Everything is determined by genes. Okay, so we have different genetic codes. Okay, some of the genetic codes that I'm talking about here is actually related to the production of protein. We say, okay, in a cell, the nucleus is responsible indirectly for making protein. How? Because it contains a genetic code that codes for a certain type of protein. Now we have different type of proteins in our body. Hemoglobin, which is a part of our blood, is for protein. Even our hair, muscles, organs, these all are proteins. So there are different type of proteins in the human body or in a plant or in any organism. So the code is going to decide what type of protein is going to be produced. Okay, this is basically a complex thing. I'm just telling you like the summary of this one. Okay, now let's one we have talked about this, the organism. The organisms are of two types. One is unicellular, other ones are multicellular. Now, what is the meaning of unicellular and multicellular? Uni means one. Mean these organisms are made up of only one cell. Multi means many. These organisms are made up of many cells. Can you give me an example of unicellular? What do you think? Which organisms are made up of only one cell? Definitely the microscopic one. It's written somewhere on this sheet. It's written somewhere on this sheet. Is my screen visible to you, EU? Okay. So it's written over here. Just find it where it's written. I'm giving you a hint. It's written somewhere. Have you found? Bacteria. Good. Okay, give me an example of multicellular organism. You're a multicellular organism. Oh, okay, it can be. There's so many. You can refer to insects, you can refer to plants. Good. You can refer to humans. You can write about any species. Okay? So it's about like you need to think think more and more about these things. Only then like it would become easy for you to understand things. Okay? Let's focus on like the things around us. Okay. Now, what is the main difference between the unicellular and multicellular else than cell, else than the number of cells? Very first thing is unicellular organism, a single cell is capable of doing all functions. Sorry, is capable of performing all functions. Okay, while in multicellular organisms there is a term called as division of labor. Like we are humans, we have different organs. We have heart, we have liver. Now it has a different function. This is called as division of labor. Okay, so when we categorize organisms, we categorize them on the basis of not only number of cells but how do they behave. Sometimes bacteria also live in a group, but still they are unicellular because each cell or a single cell is capable of performing all functions. While in multicellular organisms, many cells they combine together to perform one function. Clear? Or do you want me to explain more? One more time? Okay. Unicellular are made up of one cell, though they make a group, they can make a group, they are capable, but still one cell can be performing all the functions. Okay, one cell can reproduce, the one cell can respond, the one cell can evolve, the one cell has the genetic code. This is done by a single cell, all the functions in the unicellular organism. But in multicellular organism, the functions are distributed to different organs. Okay? Okay. Um, I guess this is all for cells. Okay, let's discuss one more type of cell. Now this is based on the number of cells, now the type of cells. This is the number of cell. Now the type of cell. Okay, there are two types of cells. Like we have discussed, one is bacteria and the other one is human. So our cells are different. How? We have a proper nucleus in our cells, right? While bacteria has no nucleus. Do you have any idea about this one? No? No. Okay. Let me give you a diagram. Okay. So you must have an idea of what do we call it? DNA. You must have an idea of DNA, right? If DNA is wrapped in something, then it is called as nucleus. If the DNA is not wrapped, okay, then there would be no nucleus. And there is a term that we use for it. prokaryotes and eukaryotes. Let me write that down. Eukaryote and prokaryote. Pro means the older one, okay? Because these one the old are the oldest one organisms that we know. So these do not have a proper nucleus. Well, if we talk about the animal cell or any plant cell, there is a proper nucleus. I'm giving you an image. Wait. Okay, here is the image. Can you see the image? Okay. This one is nucleus. So DNA is enclosed, or you can say packed inside something or a membrane, and this is called as nucleus. While if you look at this bacteria, the DNA is not enclosed, it's freely floating in the cell. Okay, so this one is eukaryote, this one, while this one is prokaryote. Clear? Okay, are you making notes? Okay, do make notes so that you can revise it later. Okay, so this is all about cell. Now let's move to the like discovery of cell. As I mentioned in the start, that there was a scientist, Robert Hooke, who actually looked under the microscope and found that these are the compartment like or the boxes like structures. So let's talk about the microscope. There are different type of microscopes available now. One is the light microscope, which is the most famous one and mostly found in our school labs. And the other one is the electron microscope, which is usually found in the high-tech lab, or maybe your school might have this electron microscope. Okay, what is the difference between these? In light one microscope, light is used as a source of viewing objects. Okay, if electrons are used for to view objects, then that is called as electron microscope. And if some kind of dye is used or some kind of color is used, then it would known as the fluorescence microscope. So we are going to learn about three type of microscopes: light, electron, and the fluorescence. And why are they named so? Their name is based on the type of source used to produce image. Type of source used to produce an image. Okay, let's talk about the light microscope first of all. What are the limitations of light microscope, or why, or what are the usefulness of light microscope? Though light microscope is used extensively, you can clearly observe cells, you can clearly observe the blood sample, the cells in the blood, you can clearly observe so many things. But the limitation is you cannot observe the processes that are going on in the organism. Like if you have placed cell or anything under the microscope, you can not tell where is the transportation occurring, or where how is protein synthesizing inside the cell. These whole processes are not cannot be explained. Okay, though. Okay, how do we say it? We use light as a source to view objects. This is point number one. Now, what does light do? Let's talk about that. Okay. Have you learned at any point of your life about light, like lights bend? How do they bend? So they can bent, right? If you place a pencil in a glass of water, you might observe it from the outside that the pencil seems to be bent, right? Now, this bending helps us to produce an image, and this bending is known as refraction. The bending of light is known as refraction, and it produces an image. Okay, the similar process happens in our eye as well. How does our eye produces an image? The light enters our eye, then get bent by the lenses in our eyes and produces an image. Okay? Similarly, the lenses are used in microscopes. The light bend and produces an image. Shall I write it here? Okay. Light bend, or we say refract, and through lens and produce an image. Shall I proceed? Okay. Okay, sometimes we use colorless images. We might not be using colorless, specimens. We might not use blood. We might use some onion, onion membrane. Let's say onion layer. Now that would be colorless. That is of white color and is usually colorless. How can we observe these colorless specimen under the microscope? Now for colorless specimen or substances, we need to use a dye. They are stained with some dye. We put a drop of dye on that cell or that layer. It gets stained. We'll let it dry for like few minutes and then observe the slide under the microscope. Shall I explain more? Okay. So what do we have? We have a glass slide like this, and here we have a layer of onion, let's say. A very thin layer. Now this is colorless. This won't be visible under the microscope because there are no colors. Now for coloring, we may use some kind of dye. A drop or two is added to this, and we'll wait for drying, because after drying it will get stained. There are certain parts which would be colored and there would be certain parts which are not stained. So we would see an image and we can tell, okay, these are the different parts of a cell. I'm giving you one diagram. Just give me a minute. This is how the stained cell looks like. Okay? Okay. Okay, sorry. What I've told you, what are the limitations? That fourth point is about the limitations. We cannot observe life processes or limitation. Well, let me just erase it. So I've changed my pen, sorry. Limitation is only cannot observe the life processes which are happening in the cell. Though we can observe many things, but not the internal, like how protein is synthesized, the very like, or how mitochondria produces energy. We cannot observe it under the light microscope. These are the name of part of cells. Don't get confused with these names, okay? I'll explain later the part of cells as well. Okay, now there is one term that we will use for all these types. That is known as magnification. Let's discuss it later because this would be applied on all type of microscope. Magnification. To magnify something, to make something bigger. Okay? Okay. Now let's talk about the light microscope or electron microscope. So as we have discussed earlier, the electrons would be used as a source of viewing object. Okay? So what is our source? A beam of electron. Now, what is the function of electrons? When electrons pass through something, they produces an image. Okay, there are certain type of lights that are used like UV light, the X-rays are used. The UV light is used. They have the different type of particles. Here we are using a beam of electron. And when this beam passes through the specimen, it produces an image. Are you getting me? Okay. Okay. Second thing is, the second point is, sorry, is point number two is a beam. Okay. Image is produced. Number three. Number three is what is the advantage of using that? We can view very even the smallest part of this one. What is not visible in the light microscope. Its magnification is much higher than light microscope. Much higher than light microscope. We can even view very small objects. We can even look into the internal part of the nucleus. We can even look into the internal part of the its parts. For example, the mitochondria, the chloroplast. We can even look into that. Its magnification is very high. This is the advantage of this one. Okay. Then there are two types of this one. One is called as scanning electron microscope. The other one is transmission electron microscope. SEM and TEM. Okay. Because this one is scanning, it produces a 3D image. Okay. The scanning electron microscope can produce a 3D image which helps to understand the cell better. For example, the bacteria. Let me show you the bacteria. The original one, more similar to it. Let me get the diagram. Okay, this one. Okay. Now here is the external and internal structure. Both are given. If we look into the external structure, the flagellum is very clear. But if we increase the magnification, we can also look into the internal parts. The magnification is really high. Okay. Another one is transmission electron microscope. Through this, we can observe the very internal parts of the different organelles. Like we can go into the details of this DNA as well. Like this one. This one. The second image is produced by TEM, which shows you all the parts. Okay. Let me check if I can get the image of posts because these are very complex one. Okay, I've got one thing. Uh, Let me show you. This is how your blood would look like under SEM. Okay, this is the 3D image. Okay, this is produced by SEM. Have you got it? Scanning electron microscope. Wait, I'll write. Scanning electron microscope. It produces a 3D image of any specimen or any sample. While if we talk about TEM, TEM shows us the internal parts. This one. This one is shown by TEM. Transmission electron microscope. Shall I show you something more related to it? Though I've shown you bacteria, this diagram is similar to that. Let me just erase it. This is the TEM and SEM. This is the 3D image and these are the internal parts. Yeah, the second one is it's showing the internal part. Look, this one must be a nucleus. Then there are certain organelles around. Okay. Okay, let's move to the third one, which is the fluorescence microscope. Wait, let's move to the fluorescence one. Okay, we have discussed earlier that fluorescence would use some type of color. Have you ever seen the UV light? You can easily check your fingerprints under it. It's a kind of fluorescence. It produces a something greenish color. So we use basically the fluorescence dyes over here. We use colored dyes to observe the specimen. Specimen is anything that we observe under microscope. Okay. Okay, why do we need to use the fluorescence one? Okay, let's just observe it. Okay. Um, Scientists want to know how water moves through the plant. What is the movement? What pathway it takes? So definitely they are going to use some color thing in water and then they will observe under the microscope. Okay, this, if they're highlighting the certain points, they would definitely know, okay, this one is showing the color, so water is moving through this path. Have you got it? No, I don't think so. Okay. If we want to check the movement of water in the cell, okay, so we would add a little dye in the water. Okay, now from whatever part water passes through, that part shows color under this microscope. So you would know the path, the actual path. Let me show you the different colors. uh Wait. Okay, this one is cool. Let's use this image. Okay, this image is produced by a fluorescent microscope. Where, from where, whichever part the dye is moving, that part would be colored only. The rest part won't be colored. So this can easily tell that how the process is actually occurring or what pathway a certain substance is taken. Okay, let me give you some other example. If you have taken an any type of medicine, it moves through your digestive system and then move to certain path of your body, right? Okay. Now, what scientists want to do when they like when they are inventing a new type of medicine, they check it on certain organisms. Now, how do they check it? They put a fluorescent dye in the medicine and then give it to some animal and then check, okay, through which pathway or the which pathway is taken by this medicine to reach that certain organ or to target the affected cell. This is why we use the fluorescent dye to check the whole process, like how the process is actually occurring. Okay. Okay, these are three type of images that are produced. Okay, let's move to the last thing, which is the magnification. Magnification tells you how large the image is. Okay, you might observe. Okay, look here. It's written 10 micron meter here in this diagram. Again, in this diagram, a 10 micron meter is written. Okay, this one is the size of that object. Actual size, let's say. Actual size. These one are really small. Do you know how many zeros are in micron meter? 6 zeros. Okay. So we have 1 centimeter. How many millimeters are there in 1 centimeter? 10. So we should know these simple calculations. Okay. These are not that much hard. Okay. The next calculation is nanometer. How many zeros would be there in nanometer? 9 zeros. Mean if we want to observe, then 1 centimeter is the largest one, then then millimeter, then micrometer, and last one is nanometer. This one is the gray, like the size is 1 centimeter would be the largest one and 1 nanometer would be the smallest one. Largest and smallest. Viruses are the smallest one. They're really small. Shall I give you the question now? Okay. Do you want to ask any question? Okay, let me give you the questions. Okay. Okay. I missed one thing. That is the division of cells. How are cells divided? Any idea how are cells divided? Okay, wait. Because we have discussed this point in the start and I have totally forgot to explain this point. Okay, how do cells reproduce? Okay. So we have written a point that new cells arise from pre-existing cells. Whatever cell exists before, right? So if there is a bacterial cell like this, is grow, grow, sorry, it grows bigger in size and then divides into two. Okay, this is the simple division. So that's why we say the new cells arise from the pre-existing one or come from the original one. One cell divided into two. Now how do they do? They split into two cells. Okay. Do the number of cell increases by this? Great. Okay. Uh, can you give me a summary of what we have learned today? Then I'll give you the quiz because I just want to know um, at what level you are. Okay, okay, let me ask questions. Let me ask oral questions to test. Okay, what is a cell? Okay. What are the prokaryotic cells? Okay, if you want, okay, and what is the characteristic of the prokaryotic cell? It does not contain any nucleus. So this is the actual definition. Okay, and what are different type of microscopes? Okay, okay, okay. The last one is the fluorescent one, which uses color. Fluorescent, fluorescence, that is related to color. Okay. Um, I hope you're going to continue this one because in the next topic we are going to learn about the different parts of cell. Okay. Okay. I'm giving you questions. Your question number one is, shall I post that in group or tell you here? Here? Okay. Okay. Your question number, let's write then. Question number one. What is cell theory? Question number two. Which one is better and why? A. The light microscope. B. The electron microscope. Question number three. How do we categorize the unicellular and multicellular organisms? Okay, you have 5 minutes to answer me this and send me answer in the chat. Okay, great. So I'll be waiting for you. I'm giving you 5 minutes. Okay. Okay, let me check. Okay, I've sent you one question. Though I'm going to paste it here as well. I just want you to go through this question and tell me what are like your correct answers. Okay, what you have written? The light. Yes. Um, what is cell theory? It's a basic unit. Oh, cell is a basic unit. It's okay. There are. Wait. The first one is okay. We're making notes. Okay, there are three main points of cell theory. You need to tell all three. Okay, you in the first one question. Are you getting me? What you need to write over there? Okay. Okay. Number two is correct. Okay. Number three is not only the number of cells. I've told you, it's based on how much a cell is capable of performing a function. In the unicellular one, a single cell is capable of performing all the life processes, while in multicellular organism, there are number of cells which perform a certain function. Okay? So answer number one and answer number three needs a bit of tuning. They need bit improvement. Okay? Otherwise, your concept have grown better. You got the point, but yeah, is capable of performing all the functions. Got it? Okay. Have you seen observed this diagram? Sorry. A number of cells or a group of cells perform a certain or specific function. Okay. Okay. Have you gone through this diagram? Okay. Is my screen visible? Okay. There are two diagrams given. A micrograph. These are observed under the microscope. That's why they are called as micrograph. Okay. Which one do you think is a eukaryotic cell? B. It contains a nucleus. Okay. And what evidence are you going to give for this one? What evidence? There, you have to choose two options. Yeah, I'm giving you time. Yes. Here. Okay. A is correct. No, because the prokaryotic ones are the one, or or the unicellular one. Prokaryotic are usually unicellular. Okay. Yeah. Okay. Let's learn more about it. If you want to have a next class, then we'll learn more about it in the next class. Okay. Uh, can you give me a break of 5 minutes? I'll just go and get water for myself. Okay, thank you. So I'll make a call again. Okay. Yes, you can. Thank you. बस हो गई थी मेरी. डेट क्या है? 3rd Feb 2026. मैं अगर तेनों भी मैं ਇੱਥੇ ही रखा ना जरा. सही है ना फ्रान? डोंट टेल देम और ज़्यादा. मैं फिनिश करवा सकती थी इसे. हाँ, मैंने नहीं करवाया. हाँ, 3rd Feb. Copy. least यहाँ पे लिखना है 740 to A40 done. मैं ज़रा let's start at 840. Hi, so I'm back. Have you taken your break? Did you drink some water? Because we need more energy for chemistry. Okay? Do it some kind of sweet. Any sweet thing. Just have a bite of something sweet.